Thank you so much. Hello, everybody. Thank you for inviting me to this seminar. I'm very glad to be part of this academy program. My name is Alexandros Spyridonidis. I'm from Patras, Greece, and we're going today to discuss about regulatory T cells in hematology. Where do we stand? As you all know, T-regs are part of the physiological repertoire, and the T-regs play a major role in controlling immune system, aiming to control excessive immune responses by suppressing a variety of immune cells. The T-regs, in my opinion, they work like the over-voltage protection relay in the electrical system. We have a lot of T-regs in the body, like natural T-regs, which are derived from the thymus, the well-known FOXP3 positive CD25 high T cells. But also we have peripheral T-regs, which are physiologically in vivo induced in the periphery. And we also have induced T-regs, we can produce ex vivo by converting T cells, conventional T cells, to T-regs. T-regs have multiple mechanisms of actions. They target many immune cells, but then they work with multiple molecules, like they can consume IL-2 through CD25, or they express inhibitory molecules like TGF-beta, adenosine, or also they can mediate, they can act through CTLA4 mediated phagocytosis of CD80-86 on the dendritic cells. As such cells are very powerful also for adoptive cell therapy. We're trying to use it in the clinic as adoptive cell immunotherapy. The most well-known cells are the natural T-regs. We're trying to isolate it through selection of CD25 high cells. The problem with this isolation is that we cannot have very pure T cells because the FOXP3 molecular is intracellular marker, so we cannot use it to isolate the cells. We use the CD25 marker, which is also expressed in activated cells. That's why at the end we don't have so pure cells. That's why we're trying to find other T-regs in the body. We can either expand the natural T-regs either in vivo with low dose interleukin-2, or ex vivo expand the natural T-regs also with interleukin-2, or we're trying to convert T cells in induced T-regs either in vivo or ex vivo. So there are many approaches in order to use them in adaptive cell immunotherapy. And of course, the most important for us, we are dealing with transplant, is to use them in order to treat or to prevent graft-versus-host disease. We all know that graft-versus-host disease is a T cell mediated disease, and T-regs are missing there, and that's why we can use them to prevent graft-versus-host disease. And I'm going to focus today on this approach, how we can prevent graft-versus-host disease with the T-regs. The first studies were done on this for this prevention of GVHD with T-regs was done for the group of Perugia, where they used freshly immunoselected CD25 high positive natural T-regs. In haploidentical transplantations, they have done up to now 3 studies. They included up to now nearly 100 patients in 3 studies up to 2021, where they isolated these T-regs. They gave it day -4. And before a transplant, they gave CD34 selected graft together with some conventional T cells. And these studies up to now showed that this approach is feasible, and maybe it can also prevent GVHD in these haploidentical transplantations, and with very good results. But as I told you before, the problem is that these cells are not so pure, and so we don't know exactly the results if these results are because of the T-regs. A better approach with more pure cells of T-regs is from the Stanford group, where they purify these T-regs not only by CD25 selection, but after the selection they use GMP flow sort to isolate the CD127 low fraction, which is for the T-regs. And they've done up to now multiple studies, phase 1, phase 1b, phase 2, and they went up to phase 3 study, which is now ongoing with a product which is called ORCA-T, where they use these free T-regs. They give these T-regs day 0 together with CD34 positive selected graft, And at day 2, they gave also some conventional T cells. And as you see, after transplantation, they use single agent prophylaxis in order to prevent GVHD. The results up to now are very promising. As you see here in phase 1, phase 2 studies, the non-eruption titer is very low, and also the GVHD, greater 3, or chronic GVHD, is below 5%. And here are the corresponding curves. We're awaiting the results of phase 3 trial, which is now ongoing, the ORCA-T trial, which is the biggest trial now for T-regs application for preventing GVHD worldwide. Of course, besides this, there are also other approaches, smaller for other groups. For example, the Blaser group, they use induced T-regs, where they produced it with TGF-beta, and they used it in phase 1 study, or the so-called tier 1 cells from Garol, also at Stanford. These are small studies, phase 1, phase 2 studies with small inclusion of patients up to now, but they're very promising results to prevent GVHD. Such induced T-regs or T-regs also used to treat chronic GVHD, like steroid-resistant chronic GVHD. You all know the paper from New England Journal of Medicine where they gave low-dose interleukin-2 in order to in vivo expand the natural T-regs with some excellent responses in patients with chronic GVHD. And this group also continued with another study where they combined the low-dose IL-2 with T-regs that they produced ex vivo, or other groups also they use T-regs in order to treat very difficult steroid refractory chronic GVHD. But as up to now, these are only small reports with very few patients, and they couldn't manage to come up to now in the clinic, but studies are there. What I'm going to discuss with you the next 5 minutes is our approach in Patras. In Patras, we have a bone marrow transplantation unit, University of Patras. We have a donor center. And we wanted also to use cellular therapies for our patients. That's why we established the GMP lab, the Dimitris Lois GMP lab in the Institute of Cell Therapy. And also we have a master program of cell and gene therapies. And our program to prevent GVHD, first of all, includes we're using in vivo T-cell depletion with, up to now, we used low-dose Cambath, as we see here, with some very good results. We did some pharmacokinetics to see how much low-dose alemtuzumab should we use for our transplant protocol. And what we are doing after transplantation in order to prevent relapse, we are using prophylactic TLI. In our first phase 1 study, you see here that when we give prophylactic TLI, we have 0 relapses. This is very few number of patients, but without prophylactic TLI, we had relapses. And we continued this approach in many patients, and we published now more than 180 patients. When we use prophylactic TLI with low-dose Cambath, this is our platform up to now for our transplantation. But still, we cannot exclude that the patients have GVHD, and in order to treat GVHD or to prevent better the graft versus host disease, this is our protocol, what we're using until now. We're using induced T-regs. How we use it? We actually mimic the nature. We looked at the nature and we looked what happens during the pregnancy. During the pregnancy, actually, it's the best example of the immunogenic tolerance in the nature, because as you know, in the pregnancy, the mother tries to attack the fetus, which is a semi-allogenic fetus, a semi-allogenic graft. And in order not to attack the semi-allogenic fetus, the immune system of the mother, in the placenta, a molecule expressed is called HLAG. It's an immunomodulatory molecule, which is expressed in the fetal-maternal interface of human placenta to protect the semi-allogenic fetus from maternal immune attack. The HLAG expression is epigenetically silenced after birth, but may positively induced in immune cells in order to maintain immune tolerance. So we try to use this, and that's why first we looked in our patients and we looked at after transplantations, So before transplantation, you don't have any HLAG positive T cells in the periphery. You don't need them. You don't need these T-regs that express the HLAG. But after transplantation, many patients have some HLAG positive T cells. So they are expanded both in blood but also in GVHD sites. And when we isolated these HLAG positive T cells, we looked in the in vitro and we saw indeed that they're very good immunosuppressive when we use as third-party cells. As we see here, without use them, these cells as third-party, the T cells expand very well. But if we use this as third-party, the T cells cannot expand, cannot activate. That's why we then looked how we can produce these HLAG positive T cells, which are also after transplantation coming out. And we saw that if we use azacitidine in vitro, we can start from some cells in the periphery and we can produce them and produce these HLAG expressing T cells, which we call them now IgT-regs. And when we looked in this T-regs, which they express the HLAG molecule. As you see here, they are very good immunosuppressive. As you see here, this is the T cells that they are very good activated, but together with HLAG positive cells, they are not so well activated. And most importantly, these HLAG positive T cells as third-party cells are act when we also have TNF, so inflammatory conditions, as we have in GVHD sites, where they still suppress very well. In contrast, the normal natural T-regs that we use the most of the time, the thymic natural T-regs, indeed, this also they can suppress, but not so good as the HLAG positive T cells, but they don't work in the inflammatory conditions together with TNF. So we believe that these HLAG positive cells that we could produce in vitro through to produce in vitro through conversion from conventional T cells, they are better than the natural T-regs because they suppress better and they work also inflammatory conditions. And when we looked in these HLAG positive T cells, they are different from the normally FOXP3 natural T-regs. They don't express so much the FOXP3. As you see here, the normal T-regs express FOXP3, but the T cells don't express FOXP3, but HLA positive express very low FOXP3. And if we try to see how they work, how they act, how they suppress, indeed they suppress through the HLAG molecule. But if we put antibodies against the HLAG, they still suppress, but not so much. That meaning that the HLAG molecule is a molecule that is mediates the suppressive activity, but it's not the only one. That's why we try to examine what other they express, and they express other regulatory classes, other markers, we'll tell you later on. So they are not working, these cells, these HLAG positive T cells, T-regs, they're not working through HLAG alone. This is a very important molecule, but also through other molecules. And indeed, we managed these three excellent PhD students, Dionysia, Memnon, and Panagiotta. They managed indeed to bring this protocol, which was a research protocol, which is an in vitro protocol. They bring it in the clinic. They produce in small scale, clinical scale, and also GMP scale. And they looked also at the cells, and they looked at the express not only HLAG, but they express other molecules like TIGIT, PD-1, LAG3, TIM3. So they are potent suppressors acting through the HLAG molecule, which is a very good marker to isolate them, but also through other markers which are expressed during the process of development for conventional T cells. And when we looked at these HLAG positive T cells, as we call IgT-regs, this is now an ATMP, an advanced therapeutic medicinal product. This, when we looked in vitro, of course, they're very well immunosuppressive, but they also, in our collaborators in Papanikolaou Hospital, we examined them in mice, and we saw that they prevent and delay DNA-driven GVHD in mice, do not cause themselves GVHD, and also they retain their GVL effect. So they have all the attitudes that we want in vitro and in vivo to translate these cells and to bring them in the clinic. And that's why we started now in protocol here in Patras, together with Papanikolaou Hospital in Thessaloniki, in our patients when they get to allogenic transplantations for 3 weeks, for 30 days, they are in the ward. And if after 4 weeks they are in good condition, we screen them and we start the manufacturing of IgT-regs. And about day 50, 60, we infuse these IgT-regs with the goal to establish immunoregulation, which is more physiological, and to stop the normal immunosuppression. So our goal is to give the IgT-regs about day 50, 60 after transplantation, and then to stop immediately the cyclosporine that we're giving. Maybe if we saw that this works, maybe to bring it earlier in day 30, and so to try to prevent GVHD without cyclosporine, only with T-regs. Up to now, we have included 2 patients. Goal is to include 9 patients. This is a phase 1 protocol, which is now running in 2 centers. And of course, the goal of this phase 1 study is to show safety and to find the maximum tolerated dose with, as I said before, to use this T-reg cells GVHD prevention protocol for our patients. These 2 patients, especially the first one, which now is day 200 after transplantation, did excellent with no GVHD, disease-free, infection-free. He was never admitted again in the hospital. He had no problems with CMV. And we could stop the normal cyclosporine immunosuppression very, very fast after transplantation, after we gave them this T-regs. Of course, we need more patients to establish that these cells are working. So this was, just to summarize here, a very, very nice journey where we started our research about 2009 to examine these suppressive T cells that we call T-regs, and they express this HLAG molecule. And it took many years, as you see here, to go to translate in the clinic, to develop this as a drug, because this is an DMP, to write investigation new drug application, and to come in the phase 1 study, which, as I told you before, it's running in 2 centers. To summarize, our strategy in Patras for our patients is, in order to enhance the viral immunity, we use pentavirus specific T cells together with Papanikolaou Hospital in Thessaloniki. To prevent GVHD, our platform is the low dose T cell depletion with alemtuzumab, or the PTCI platform nowadays. And our goal is now to use routinely the IgT-regs to prevent GVHD. And of course, to prevent relapse, we have many protocols or many algorithms, like using prophylactic DLI, TKIs if the patient has affected 3-ITD leukemia, or included in some studies like VLT study or LNILL, etc. So this, as I told you before, was a long journey to produce these IgT-regs and to bring it in the clinic. I believe this is the first or the second study in Europe which with the application of T-regs in patients to prevent GVHD. And this is a teamwork for all this team in our hospital, especially in the Institute of Cell Therapy, and of course our collaborators in other hospital, and from the supporters and the funding that we receive from very enthusiastic people. Thank you so much, and if you have any questions, you can contact me, and I'm very glad to have in the future many centers be included and worked with us in this T-reg program. Thank you.